Yes. All right. So welcome everybody. Thank you very much for attending the seminar. As Michael has introduced us, I'm Emanuel Souza de Quadros. My background actually is in computational linguistics, and right now I work as a natural language processing researcher at the DSMZ, more precisely at our data integration team led by Dr. Julia Koblitz, which is a team that has as its overarching goal connecting the several databases that we have at the DSMZ, both between amongst themselves, but also with other external sources of data and making sure that all of these data sources are interconnected and usable in the institute and for other applications. Okay. All right, so as you've heard already, the topic of this session is the use of LLMs in bioindustry in terms of knowledge-based expansion, data integration, and insight generation. So I'm starting the session because I'm starting from these two initial components of knowledge-based expansion and data integration, such that they can feed later the topic of insight generation that will be treated more expensively by Marco in a few minutes. So in terms of knowledge-based expansion, I'm talking about integrating external data, more precisely in this case, literature into our internal data stores. Right? Of course, the expansion of knowledge base can come from different sources, from experimental data, from like the deposition of new cultures in a microbial collection, for example, or also from the research literature, and I'm going to focus on the latter. In terms of data integration, of course, we want to make sure that all of these data stores are properly connected, interoperable, and usable, especially in this case, in insight generation for bioindustrial applications, as we will see later. Focusing on this task of adding information from the research literature into our internal data sources, I want to present you, in case you don't know, like this several databases that we have at the DSMZ covering a variety of topics like RNA sequences, bacteria. More recently, we also have a plant virus database that is not on this picture, a nomenclature database, and several others, right? And many of these databases make use of information from the research literature in one way or the other, either by linking, by referencing, simply referencing that information, or by incorporating information from the research literature, which is the case of some of these databases, and specifically the case of Brenda, which I will talk more about later, which is our enzyme database. Of course, incorporating information from the literature can be a very time-consuming process that requires often a lot of manual annotation, which is time-consuming, error-prone, and very costly. In addition to that, we know that the research literature grows at a very fast pace that can hardly be matched by the pace of human annotation. So part of the goal of the project that I'm working right now and that I'll talk more about later is streamlining this process of literature annotation for use in our databases. So here I'm going to present you first an overview of the bigger picture of this process that involves and this subdiscipline of natural language processing, we can call it that, called text mining, which means going into texts that have the information that we might want in an unstructured form, and extracting that information and translating that into however, whatever structure we are interested in. This often can take the form of a knowledge graph, which is a structure that connects entities organized in a particular ontology, depending on your domain. So, for example, in the domain that I will present you shortly, we have several categories of entities. For example, bacterial species, bacterial strains, or enzymes. And so the entities that we find in a text can be organized in that ontology and connected by certain relations that are also specified by our ontology. This structured representation of the data can then be used in other applications. For example, in a decision support system that will help researchers to find insights into this structured data. That will be covered later by Michael. On this process of text mining in this case involves two main natural language processing tasks that we will talk more and that I'll talk more about. One of them is entity identification. Of course, we are interested in certain types of entities in a text, and we want to be able to identify them precisely in the text. Once we have identified them, we are interested in the other a further step, which is a step called relation extraction, usually, which means extracting which relations obtain between the entities that we find in text. So, for example, here we can see that the text supports serves as evidence for saying that ATCC 25544 is a strain of Rhodococcus erythropolis, right? The text also offers evidence that cholesterol oxidase can be expressed by this bacterial strain. And we want our text mining model to be able to extract that information from the text as automatically as possible. Okay, as I alluded to before, I will focus specifically in one project that I have been working on, which is the task of extracting strain-level metabolic information in Brenda, which is our enzyme database. Brenda in particular relies very heavily on literature references. In its current version, it has more than 170,000 literature references about describing metabolic information for different types of organisms. And specifically in this case, we are interested in the more than 50,000 references that connect bacteria to enzymes. The specific problem that we are focusing on in this universe of references is the level at which these relations are annotated. Most of them are annotated only at the species level. However, we would like to have also the information about this metabolic information also at the strain level. Why would we like that, right? So at the digital diversity hub, which you can access at this address, hub.dsmz.da, we are building this strain profiles, which are these profiles that connect information about a particular strain coming from several sources within the DSMZ and outside, right? Here we have one example of which you can find on this link for this particular strain of Corynebacterium glutamicon, which is the example that right now is shown in the website. And we have this table of enzymes that can be expressed by this strain, coming from Brenda, coming from BacDive, which is our database for bacterial information. And so we can see that we can connect like several databases through this string profile. However, we need to have for that information at the string level, right? Because it's not enough to say that this bacterial species can express this enzyme. We also need to know that this particular strain can do that. And you can see, of course, how important that can be for bioindustrial applications when you want to know the specific strain that you need to produce that particular enzyme. All right, so what we want to do naturally here is to go back to those references that are annotated only at the species level and collect information also about the strain at the strain level. But we don't want to do that manually or purely manually on thousands and thousands of references. This task of relation extraction is one that is quite difficult in terms of natural language processing. And the reason for that is that, as you might know, machine learning models require a lot of training data, right? And training data, the best kind of training data is human curated, human annotated training data, which is severely lacking in this domain. Also, you might know that machine learning models also need negative evidence to learn to code a certain type of relation as not existing, for example, for a particular combination of strain enzyme, right? Problems that negative examples are very rarely annotated, even though they are more common than positive ones. So here we have one example of negative evidence. We have this bacterial strain co-occurring in a text with a particular enzyme, but we want to have that annotation that saying that there is no relation between this strain and this enzyme, or more precisely, that this strain does not express this enzyme. Naturally, this annotation will not be present in a database like Brenda, because Brenda is showing you which strains express a particular enzyme and not those that do not, right? However, in building a data set for relation extraction, we have this advantage of having already a human curated, a high quality database like Brenda that offers us information about the relations that exist between particular strains and particular enzymes. By trusting, as long as the database, the process of annotation that generated this database was of high quality, we can infer that strains that are present in the database and are not connected to an enzyme, that have not been annotated for a particular document, we can infer that that relation was not found by the annotator in that document. So by building on a highly well-curated database, we can extract a data set that has that information. So at least we have that advantage on building a data set of relation extraction for this task. And that's what we have done. So we have translated those database relations into a text format that can be read by humans with some care, some difficulty, but very easily read by machines, right? Another challenge that we have in this sort of task, and specifically for this database, is the problem of data imbalances. As you might expect, depending on the interests of the researchers that build the database and the interests of the research community at large, we might expect several very sharp data imbalances to occur. And that's what we find in this database. So we have almost 40,000 strain enzyme relation annotations in a database, counting repetitions. And most of those annotations are linked to one single literature reference, meaning that particular strain enzyme pair only appeared once in the whole database connected to a single literature reference. On the other hand, we have a very small number of strains, 152, that account for almost 25% of all relations that are annotated in this database. Of course, we don't want a model that would be very good at predicting those particular relations and generalizing that to cases where that relation doesn't exist. So we have to deal with these data imbalances in some way. Just as a curiosity, we have here the champion with 58 mentions in the database. We have Saccharomyces sulfataricus P2 connected to DNA-directed DNA polymerase, which is the largest, the strain enzyme pair with the largest number of relations, of annotations. All right. So this is a detail that if you're interested, we might talk more about later, but not super interesting for this talk, which is there are several ways of handling these data imbalances, and in particular in this project, we are using this process of greedy maximum entropy sampling, which basically means that we add documents to our data set, to our training, validation, and test data, maximizing the uncertainty about which relations, which entities can be found in that set. In other words, maximizing the diversity of those sets. All right, so here we come to the actual model that can predict the existence of those entities and the existence of those relations in a document. So here we have this end-to-end relation extraction model. And it's called an end-to-end relation extraction, end-to-end model, because it covers these two steps of relation, of entity identification and relation extraction in a single model. So it does all of these tasks at once. Yeah. We have several components here, and at the top you can see this input embeddings being fed by this particular language model that we are using called BioLink-BERT, which is a large language model that is relatively lightweight and trained also not only in terms of predicting text like every language model, but also on predicting relations between articles in the biological domain. And this is a large language model that is responsible for generating the input embeddings that serve as input to the rest of the model. Okay? So let's zoom in a bit on the top of this architecture, on the input embeddings, and see how that works and how they are generated. All right, so one thing that is very important that allows language models to work for us in those tasks is the fact that language has a statistical structure, okay? Which is very fortunate for text mining applications. Like as speakers of English, native or not, you can probably predict at least the general class of entities that could fill these gaps, right? The strain produces both cell-linked and extracellular something, right? You can probably predict that that's not going to be a plant, right? Or even a bacterial strain. It is most likely an enzyme, right? You have access to that intuition without knowing so much about the domain, just by the fact that there's a certain statistical structure in language that allows us to have these expectations. Even if in our brains we are not explicitly computing the probabilities of those words appearing, we have very sharp expectations that, okay, cholesterol oxidase is a likely expression to fill that gap, but I don't know, water bottle is not. Language models are essentially models for next word prediction. That's basically all they do. And it's very, very interesting and to a certain extent surprising that when we use chatbots, they sometimes, not always, show what seems to be very intelligent behavior, but what they are doing in the background is basically next word prediction. So taking this example phrase here, the strain produces both cell-linked and extracellular something, a language model has to be able to compute the probability that that gap will be filled by any word in the language and find the most likely one, which will usually be an expression that makes sense in that context and sometimes even gives us the right answer that we want when we are talking to a chatbot. So in this case, for example, we want the model to give us a higher probability for cholesterol oxidase than it would give, for example, to E. coli, to Billie Eilish, and or for WHO. And of course, we also want, even though Billie Eilish is very unlikely in this context, it is more likely than WHO for grammatical reasons. At least it has the right word class to fill that gap. And that kind of information, along with meaning, is also something that language models have to be able to learn in order to generate language. All right, in order to do that, of course, words are in practice decomposed into real valued feature vectors, which are what you might see referred to as word embeddings, right? So in this, the idea is that these words are embedded in a multidimensional space of features that allows the model to compute those probabilities. We want to estimate for each word, for each token, appropriate vector representations that allow the model to make these predictions. In order to do that, the features that are encoded in these vectors or embeddings have to reflect some aspects of the meaning and the grammatical distribution of the tokens that we have in our data. And that's the secret sauce of a good chatbot. The chatbot is not understanding those meanings or in any deep way, but is using those features to compute the probabilities that a certain word will follow another, and so on. Very importantly for this project is the idea that because the parameters that are learned by the model have to reflect regularities of meaning and syntactic distribution, we can use this information, those features that are learned by the model, to predict that a word, for example, in this position, is likely to be an enzyme. Because if an expression in this case, refers to an enzyme, it will be more likely to occur in contexts where we would expect an enzyme to occur. Right? So indirectly, we can learn about these very specific features of the meaning of an expression by the fact that those meaning features are useful for predicting the distribution, the probability that a word will occur when following a sequence of words. Because of that, we can also use those representations not only to predict what word will follow that expression in a text, as a chatbot will do, but also to predict other features of that expression. For example, what class in a particular ontology that word belongs to. So we can use those features to train a classifier that will give us the probability that cholesterol oxidase is an enzyme, or the probability that it is a strain. And hopefully we'll find that the probability that it is an enzyme is higher than the probability that it is a strain, if the model is correct, right? All right, so now we can go back to that architecture that I showed you before, and I just covered the top of it, those input embeddings that are these vector representations that are learned by the language model and associated to specific tokens. Once we have those representations, we can feed those to a larger to another neural network, which is in this case is composed of these other components. And we have some hidden layers that will learn more specific parameters for those words. We'll fine tune those representations to our task. And then we can feed those representations to a number of classifiers. In this case, we have a class classifier, which will give us the information of whether a token is, for example, a bacterial strain, or just a bacterial species, an enzyme, or if it's not any of them, right? We also have an entity classifier that will give us the information of whether this token represents one particular entity that we know about, for example, cholesterol oxidase, or if not, if it's just another regular word, or maybe if it is an entity that we might know the class to which it belongs, we might know it is an enzyme, but we might know which specific enzyme it is. That's also important information, right? We might have an enzyme in an article that, although it is an enzyme, we still don't have it in our data source. Once we have done that, we can feed those entities that were found by the model to a relation classifier, which will score each pairs of entities according to whether they can be said to represent a particular relation. For example, the relation of having that enzyme or being able to express that enzyme. Or another relation that can be detected by this classifier is that a particular strain is a strain of a particular species. Okay. Once we have those predictions made by the model, they can be reviewed and they usually need to still be reviewed by human annotators, because machine learning models, although they are very good, they are not perfect, and we want to have data sources of high quality. We still need them to be, those results to be reviewed by human annotators. But the good news is that once we have that system, this review process can be much faster than earlier, when all of the annotation had to be made by hand, right? And so we have built this digital diversity annotation hub that is currently under test at the DSMZ, where annotators are given the predictions of the model and can reject those annotations, accept those annotations, or edit those annotations. This review process, of course, will feed back into the database and generate additional training data for the model. So the model keeps improving from the results of the review process at the annotation hub. So we have this cycle of human-machine collaboration, so to speak, where we build a text mining model with information from the databases, from the literature, and possibly from an ontology, if that's available. And then we generate predictions that are revealed by human annotators, go back to the model, and also go back to the database. Finally, once we have this structured data collected by the model, this information can also be fed to another step, which is for example, a decision support system to help researchers generate insights from that structured data. And that's where Marco will pick up in a film shortly. So thank you.